Today we're going to learn about meiosis, but before that we are going to learn a bit about crossing over first. So what is crossing over, right? Crossing over. So crossing over is the exchange of genes between homologous pairs. Now what is homologous pairs of chromosome? So before that, let's say this is a chromosome, typical chromosome, and these are the sister chromatids. So these are sister chromatids of a chromosome, and these chromatids are attached at the center with a centromere. Now we know that human cells contain 23 pairs of chromosome, right? We have 23 pairs. And each pair contains a chromosome from mother and another from father. So one comes from mother and another one comes from father. So let's say this is a chromosome from mother. Okay, and this is a chromosome from father. And this chromosome pair is our homologous chromosome pair. So this is a homologous chromosome. So the chromosomes in a homologous pair will be similar in size, shape, structure, and their genetic content, etc. Now we know that there are genes of various traits that are present throughout a chromosome, right? Like genes for our hair color, genes for our eye color, our height. So let's say these are genes for specific traits. Okay. Now there are a lot of genes present throughout the chromosome, but we are going to point out just two genes for a better understanding. So we can see that these genes are present in some specific locations within this chromosome, right? Now these locations are called loci or locus in singular form. So they are called loci. These locations are called loci. So the genes within a chromosome present in a location called loci or locus in singular. So now let's say, for example, this locus here contains gene for our hair color. Okay, so let's say this is a gene for our hair color. And this locus contains a gene for our eye color. Or let's say height. And let's not say eye color, let's say height. Okay. So what happens in a homologous chromosome pair is that this same gene will be present in this chromosome as well in the same exact location. So the gene for hair color is present in this locus as well as in the locus for this chromosome. It's the same location. The same goes for the gene for height. They will be present in the same location, the same locus of the second chromosome in the pair. So in crossing over, we can see that this gene, the gene for hair color, has two copies, right? This is one copy and this is another copy. And we call these copies alleles. So this is one allele of the gene of hair color and this is another allele of the gene of hair color. So a particular gene can contain one or more alleles, okay? So here we have two alleles for this particular gene, which is hair color. The same goes for height. We have two alleles for the gene for height. So now let's say this allele for hair color is black, okay? So we're going to write it as capital B. Let's say the hair color is black. This allele for the hair color is black. And this allele is also black. So we can see that these two alleles are identical, right? So when two alleles of a particular gene are identical, we will call them homozygous alleles. So when they are identical, we will call them homozygous allele. However, let's say this allele for the gene height is tall. Let's say this is tall, okay? This is tall. So we will write it as capital T. And let's say this allele for the gene of height is short, okay? So we're going to write it as small letter t. Now we can see that these two alleles for the gene for height are not similar to each other. They are not identical. So when two alleles of a particular gene are not identical, we will call them heterozygous allele. So when they are not identical, non-identical, they will be called heterozygous allele. Non-identical, heterozygous allele. So homo means the same and hetero means they are different. Now in the case of crossing over, what happens is that the alleles get exchanged between these two chromosomes, okay? So what's going to happen is that this allele will get exchanged here and this allele will be exchanged here, okay? The same goes for the gene for height. So in that case, this allele will go here, will get exchanged here. So in that case, it will be capital T in that case, right? So now it is capital T. And this is short letter T. This will be our short letter because it has got exchanged. So now it will become short. So the genes or the alleles of the genes have been exchanged within this two chromosome in a homologous chromosome pair. So this causes genetic variety within organisms. Okay, this is the reason for genetic diversity among us. So this was basically what crossing over is, and now we are going to move on to meiosis. So after crossing over, is a cell division process in which the formation of gametes take place. So by gamete we mean egg cells for females and sperm cells for males. So meiosis is also called reduction division. So a meiosis division process is usually divided into two stages, which are meiosis 1 and meiosis 2. Each stage is subdivided into 4 phases. So for meiosis 1 we have prophase 1, metaphase 1, anaphase 1, and telophase 1. Similarly, for meiosis 2 we have prophase 2, metaphase 2, anaphase 2, and telophase 2. So first, let's start with meiosis 1 stage and the prophase 1 phase of meiosis 1 stage. So let's start with meiosis 1. Prophase 1. So prophase 1 is further subdivided into 5 phases, which are leptotene, zygotene, pectine, diplotene, and diakinesis. So at first, let's say this is a cell. Okay. This is a cell. And within our cell we have a nucleus membrane, and within the nucleus we have these chromatins, coiled chromatins. Okay. So during leptotene phase, these chromatins, DNA replication will take place, and these chromatins will condense into thick chromosomes. Okay, so now we can see that the chromatins have condensed into these chromosomes. These are chromosomes, and this happens in the leptotene phase of prophase 1. So after leptotene, these chromosomes will form homologous pairs. So say this is a chromosome from mother. This is a chromosome from father. Okay. Similarly, we have another homologous chromosome pair here. So let's say this one is from mother. And this one is from father. So this is a homologous chromosome pair, right? There are two pairs of homologous chromosomes. So this is one homologous pair and this is another homologous pair. Now note that these two chromosomes are not homologous. They are heterologous chromosomes because they are not within the same pair. They are in a different pair. So we will call them heterologous chromosomes. But these two chromosomes and these two chromosomes, they have together formed a homologous chromosome pair. And the formation of homologous chromosome pair is called synapsis. So this formation is called synapsis. Okay? And this whole thing takes place in zygote phase of prophase 1. So this is zygote phase. So after this, the formation of homologous pair, we will see that the chromosomes within these homologous pairs have attached with each other. So if this was the chromosome from father, this was the chromosome from mother, right? So we can see that the pairs have attached together with their chromatids, right? The chromatid of this chromosome and the chromatid of this chromosome have attached together. And this happens in the pectitin phase of prophase 1. So this is the pectitin phase. So at the location in which they are attached is called chiasmata. Chiasmata is in singular form, and in plural form it will be chiasma. So these two locations together will be called chiasma, but in singular term it is chiasmata. And the formation, the attachment of these two homologous chromosome is together called tetrad. So this is a tetrad. It's called tetrad because we can see that there are 4 chromatids within this formation, right? So 1, 2, 3, 4 chromatids. So therefore it is called tetrad. And when the formation of tetrad has happened, the crossing over will already start to take place. So this was pectitin phase, and after that we will move to diploid phase of prophase 1. Diploid phase. In this phase, the homologous chromosome pairs will start to move away from each other. They will start to move away from each other. However, they are still attached at their chiasmata, right? So they will instead form a loop in the middle. So we can see that there's a loop in the middle. They're starting to move away from each other, but they're still attached at the chiasmata. So in that case, they will form a loop in the middle. And this happens in the diplotin phase of prophase 1. So after diplotin, we will move to diakinesis. We'll move to diakinesis. In diakinesis, the homologous chromosomes will separate from each other. So here we will see that so here you can see that the homologous pairs have separated. There are no chiasmata anymore, right? And in this case, the crossing over has already finished. So we're going to see that some genes have been exchanged. Genes have been exchanged after crossing over. So we're going to see there have been some exchange of genes after crossing over. So we can see that due to crossing over, these genes have been exchanged between these homologous chromosome pairs, right? And at the end of diakinesis in prophase 1, the nuclear membrane will also disappear. So our nuclear membrane will disappear in this phase. So with this, prophase 1 will come to an end. And now we will move on to metaphase 1. Phase 1. So metaphase 1 is kind of similar to the metaphase of mitosis division process. So in this case, our centrioles will move to two different poles, and they will form spindle apparatus, and the chromosomes, the homologous chromosome pairs, will move at the center of the spindle apparatus. The center is called the equatorial line, and the homologous chromosome pairs will move to the center. So let's say So we can see that the homologous chromosome pairs has moved to the middle. And after this, we will move on to anaphase 1 phase. So we will move to anaphase 1. Okay. Now in anaphase, the homologous chromosome pairs will separate from each other and they will move to two different poles in the spindle apparatus. So here the chromosomes will move to two different poles. Now this is unlike the anaphase of mitosis process, because in mitosis process during anaphase we see that the centromere gets divided in the middle and the chromatids move to the different pole of the spindle apparatus. But in this case, the each chromosome in a homologous pair will move as a whole to two different poles of the spindle apparatus. So we can see that the chromosome, there is a whole chromosome. These two are whole chromosomes and these two are whole chromosomes, and they have separated from their homologous pair and moved to different poles of the spindle apparatus. So there is no division of the centromere in the middle, right? So after anaphase, we will move to telophase. And in the case of telophase, the spindle apparatus will disappear. And nuclear membrane will start to reappear. And a cell plate will form in the middle. So now we have two daughter haploid cells, right? Now we call this haploid cells because we can see that the chromosome number is now half of the chromosome number at the beginning. So at the beginning we had 4 chromosomes, and now in these two daughter haploid cells we have 2 chromosomes in each. So this is why we call mitosis a reduction division process, because the chromosome number gets halved. So with this, the mitosis 1 stage comes to an end. So now we will move to meiosis 2 stage. And the meiosis 2 stage starts with prophase 2. So prophase 2 is, the whole meiosis 2 stage is basically the same as mitosis division process. So in prophase 2 of meiosis 2 stage, the chromatins will condense into chromosomes, and the nucleolus and the nuclear membrane will start to disappear. And in metaphase 2, spindle apparatus will form, will be formed. And the chromosomes will position themselves at the center of the spindle apparatus. So this is metaphase 2. And then in anaphase 2 of meiosis 2 stage, the chromosomes will move. The centromere will be divided in the middle, and the chromatids, the two chromatids of the chromosomes, will move to the different poles. So here we can see that the centromere will be divided in the middle, and chromatids will move to two different poles. So this is similar to anaphase of mitosis, right? The centromere is getting divided here. The same will happen in this cell. The centromere will be divided in the middle, and the chromatids will move to two different poles. So here we have this chromatids in this pole, and these chromatids in this pole. So this is anaphase, and after anaphase 2, we will move to telophase 2. So in telophase 2, the spindle apparatus will disappear. And the nuclear membrane will start to reappear surrounding the newly made chromosomes. So here, the nuclear membrane will reappear surrounding these chromosomes. So as a result, we can see that there are now 4 haploid cells at the end of meiosis 2 stage, and each cell contains 2 chromosomes, which means they are all haploid cells because the chromosome number has been halved. The chromosome was 4, number was 4 at the beginning, and it has been halved to 2. So therefore they are called haploid cells. Now let's say these 4 haploid cells, or these 4 gametes, these are gametes, okay? So these 4 gametes, let's say they are egg cells of a female, and similarly sperm cells from a male will be produced in the exact same process. And let's say this is a egg cell, a gamete from here, one gamete from here, and we have 2 chromosomes in this egg cell. Let's say this is an egg cell, and this is a sperm cell. This one also has 2 chromosomes. Together, the sperm and the egg will form a zygote, and the zygote will be diploid cell. Because the chromosome number is now back to the same number as the beginning. The chromosome number here is now 4 again. So this is a diploid cell and we call this zygote. So this is our mitosis process, and mitosis division process is a very significant division process because of meiosis we get genetic diversity. So because of this genetic variety, each human is different from another human. Like we can see that children are different from their parents. They are even different from their siblings, right? This is because of genetic diversity that happens due to crossing over, and crossing over takes place in meiosis process. Also, meiosis helps to prevent the doubling of chromosomes in each generation. So we just learned that meiosis is a reduction process, right? So in meiosis, the chromosome number gets halved during the process, and at the end of the process the chromosome number gets back to the number at the beginning. So this way the numbers don't get doubled in each generation, and the numbers remains constant for each generation. So this was meiosis process, and we are going to end today's video here. So thank you for watching and stay tuned for the next video.